Now, and Laurel, you have 75 minutes.
Okay, thank you very much. Let me just see if I can share this. Okay. Okay, so thank you very much for inviting me here today. Thank you for tuning in to listen. So as Peter said, I'm Laurel Fogarty. I'm a senior scientist in the Max Planck Institute for Evolutionary Anthropology. I'm an evolutionary theorist, and I mostly focus on non-genetic transmission and its interactions with genetic evolutionary processes. You're going to get my most speculative, my most work in progress project today. But I want to tell you a little bit about my background and the lab that we run here, just to sort of set the stage for the project and the premise of the project, why we're interested in this work, why we do the kind of work we do, and what kind of insights we hope to sort of generate from doing this. So I work in the Max Planck Institute for Evolutionary Anthropology, and there I run the Theory in Cultural Evolution lab along with my colleague Anna Candler and Adam Powell. And our lab has, so you had a talk from Anna recently, so you probably know a little bit about the kind of stuff that we do. But the lab as a whole has a couple of aims and sort of grand aims. So the first one is to expand and kind of fortify the theoretical foundations of cultural evolution. And to do this, we have a lot of kind of pure theory projects. So here we're dealing with, really fundamental foundational questions of how culture changes over time and how we can model and describe those processes. And we also want to provide some guidance on using theoretical models. So on using theoretical models or theoretical cultural evolution and to generate inferences and information about cultural data. So to do this, we have a lot of projects that try to connect data, cultural data in particular, to the theory of cultural evolution. So the project that I'm going to talk to you about today kind of spans both of these aims. I'm going to focus mainly on the theoretical, the analytical theoretical aspect of this project, mostly because I haven't finished the other part yet, but I will talk in broad strokes about what we hope to achieve in the sort of data analysis section of this project as well, and sort of later on and in very general kinds of detail. Okay, so I understand that this is quite a diverse audience, mainly game theorists. I'm not going to talk about game theory, so I'm very sorry about that, but just want to give you a few definitions for a diverse audience so that we're all on the same page, so that you know what I'm talking about, and everyone can follow the models and everyone can understand what I'm saying and I don't get too confusing. Okay, so firstly, just at the very base of all of this, when I talk about culture, I'm referring to kind of a collection of cultural traits that are more or less discernible from one another and that are learned, so by any process of non-genetic transmission. So sometimes it matters a lot which transmission process we're talking about. In the case of this model here, it doesn't matter so much. So we're talking about, cultural traits that are learned and maintained in a population through, imprinting, observation, imitation, direct teaching, any of these processes. The important thing here is that, cultural traits are maintained socially, so they're socially transmitted. Sort of on the back of that kind of definition, cultural evolution assumes that culture changes through processes of innovation, selection, so cultural selection and natural selection, and drift of cultural traits. And this is sort of an analogy to genetic evolution, which, where the frequency of genes change through processes of mutation, selection, and drift on genetic traits. And we assume that the tools and methods and theories developed by evolutionary theorists can be adapted to account for the idiosyncrasies of cultural transmission and used to describe cultural change. So we hope to gain some insight into how cultures change over time in this way. And the last part, and this is really what I'm going to be talking about today, is gene culture coevolution. So putting all these things together, gene culture coevolution is a branch of theoretical population genetics, which aims to understand the effects of transmission of both genes and cultural traits from one generation to the next, and particularly the interaction between those two. And we're most concerned, and certainly I'm most concerned, with cases where culture is able to change the transmission of or selection on genetic traits, so genes. less interested in the converse, which is more of a behavioral ecology or sociobiology type of focus, which I don't tend to have. Okay, so that's the gene culture coevolution definitions. And the last definition that I want to give is for a rescue. So I want to talk about gene culture coevolutionary rescues, population rescues. And population rescues happen when adaptive evolutionary change and restores a positive growth rate to a declining population and prevents extinctions or extirpation, so local extinctions, rather than full species extinctions. And population rescues have well-described population level demographic patterns. So they're very recognizable if you have time series population size data, for example. And I'll come back to that in a couple of minutes. And I just want to say, you know, there are lots of different types of population rescues. I'm only going to talk about one type of population rescue, and that's an evolutionary rescue. So the evolutionary rescues focus on rescues that happen as a result of the emergence of new beneficial mutations for adaptation. So there are lots of other ways to adapt through standing variation, through migration, etc. Lots of different ways to have a population rescue, but I'm only interested in this one very specific and maybe rare type of population rescue today. Okay, so that brings me very rapidly in 8 minutes to the premise for this project. So the premise for this project is that, you know, undeniably humans are very demographically successful species. So we moved out of Africa, we moved all around the world. We now live permanently or semi-permanently on every continent on the planet. And often, so if you read cultural evolutionary papers or social learning papers, the first paragraph usually contains something about how successful we are and how this is because of our cultural capacity. So we sort of understand in some way, intuitively or otherwise, that our cultural abilities can somehow allow us to adapt to a really staggeringly wide variety of harsh environments, and maybe even save us from local extinctions. But we understand a lot less about how technically that plays out. And most importantly, for the sort of ultimate aim of this project is, what does that look like in our genes? So if we take human genetic data and we look into human genomes, could we see these processes playing out, and what would those signatures of those processes actually look like? So one of the sort of important questions to ask maybe at the start of a project like this is, you know, do rescues or have rescues actually played an important role in human evolutionary history? It's very hard to know whether that's true or not a priori, because of course we don't know what the patterns look like, so we don't know what to look for when we look for them. But we do know a little bit. We can glean a little bit of information about this. So one of the best lines of evidence that we have on this is not from genetic data, but from cultural data. Primo and Kuhn in 2010, these are two archaeologists looking at Paleolithic stone tool culture. They suggested that a pattern of local extinction and recolonization might have been common in particularly the Paleolithic, and that it's consistent with patterns of material culture change. Whether that is what actually happened or not, I mean, I don't know, they don't know, we don't know, but there's only one way to find out, and that is to find out what the processes look like and how we can discover evidence of them. Okay, so I'm just going to dive right into rescues if no one has any questions on that stuff. Okay, so as I said, population rescues have, you know, very distinctive demographic patterns. And so I'm going to just look at one of those now. So along the x-axis here, you're seeing time. On the y-axis, you're seeing population size. So the population is going to start at a size of 1,000. I chose that number arbitrarily. It doesn't mean anything. But the population is going to happily fluctuate around 1,000, so around some sort of carrying capacity, until it encounters a lethal environmental shock. So by a lethal environmental shock, what I really mean, technically speaking, is that the population growth rate falls below replacement as a result of a genetic mismatch between the environment and the background genetics. So this is what happens, you know, in the absence of any evolutionary response to such a lethal environmental shock. The population size declines and continues to decline, and eventually the population will go extinct. So what are the ways in which we can stop that from happening? So as the population declines, at some point, perhaps a beneficial mutation arises. So this mutation is now beneficial in the new environment, and it restores positive growth rate to the population. So as this sort of spreads through the population may still continue to decline, but at some point, the population growth rate is restored. The population reaches its original size, and this is a population rescue. So it has this sort of characteristic U shape. And we can track the frequency of the beneficial value.
Sorry,
yes.
Is it important that it comes back to the original size, or could it just reach a lower size? And would that be okay? Would that be still a rescue? Yeah,
I think that would still count as a rescue. Yeah, that it just sort of, stops declining, perhaps, something like that.
Thanks.
Yeah. So you can also track the frequency of the beneficial allele as we go through this process. It also doesn't have to reach a frequency of 1, but it might do. So one of the important things that you'll notice, of course, is that there is a period of time between when the environmental shock happens and when the mutation arises, the beneficial mutation arises. This is known as the waiting time for a beneficial mutation. So one important thing to note here is that there's been, a fair amount of work done on this process, and most of the work has been done sort of in the context of bacterial populations or viral populations or populations of insects. But in general, large-bodied mammals are not thought to be able to undergo a process like this. So for example, for humans, the waiting times for beneficial mutations is likely to be very long. We have very long generation times, relatively small populations compared to, for example, a bacterial colony. And we have low germline mutation rates. So even compared to closely related species, even compared to chimpanzees or orangutans, we have very low germline mutation rates. So evolutionary rescues like this specifically are kind of unlikely to occur in mammals in general, in primates in general, and probably in humans especially. So just as a quick disclaimer, in case anyone's upset, there are lots of other ways for humans to adapt to changing environments, of course. They can adapt from standing variation, they can adapt through migration, and of course mutations arise that are beneficial to us all the time. But this particular possibility in this particular scenario is unlikely to work out for humans. But then we need to ask the question, how would a human population actually adapt to an environmental shock? And I think it's fair to say that humans might well adapt culturally before they adapt genetically. So this is the kind of thing that we're hoping to model. So again, if we look at this lethal environmental shock this time, instead of looking for a beneficial mutation, we look instead for a beneficial innovation. So the beneficial cultural innovation arises here, and then instead of the population dying out, declining, as it would in the absence of this cultural innovation, maybe it just continues to die out, but a little bit more slowly. And this gives us the breathing room for beneficial mutations to arise and then rescue the population in the way that we looked at before. So we can track, again, the frequency of the beneficial innovations, and then we can track the frequency of the beneficial alleles later on. So, okay, how do we model this? So the first thing that we want to do is zero in on the correct questions to ask. So the first thing that we want to ask is it actually feasible under reasonable parameters for gene culture interactions like this to rescue a declining human population? So is it feasible for culture and gene culture interactions to open this seemingly impossible route to adaptation just to humans, so not to other primates, just to humans? If it is feasible, yeah.
So you say you call this gene culture interactions, but the process that you just described is just cultural, right? I mean, if they would die out, it would have an effect on the genes that are there, but there's no change in genetic makeup, right?
So I will get to the intricacies of the model in a couple of minutes. The culture is, the way that I've modeled it, at least, the culture is largely independent of the genetic makeup, but the fitness of the genotypes is influenced by which cultural trait the individuals possess. So there is a gene-culture interaction in the model.
Can I add something to this? I mean, I think what you said before was that it gives you time, right? This is the crucial thing, right? That culture, that if you have this new culture now, then that gives you time for a genetic mutation to occur. And then that
it gives you time, yes, but it also affects how both the gene and the culture spread. So I'm going to get to it when I come to the results, but the way that the genetic allele spreads is influenced by the presence of culture once it arises. So there is an interaction there, yes. Maybe it'll become clear when you see the equations. Okay, so yes, we're interested in exactly how we could model such a process. What would it look like? And of course, as we talked about before, whether we can detect it or not. So we take the lead from existing evolutionary rescue models. So there's two that I like in particular that I have kind of leaned on quite heavily, Osmond et al. In 2019 and Osmond and Coop 2020. So these are obviously purely genetic evolutionary rescue models where the population is rescued by a selective sweep. We want to add culture to this and add an interaction between the genes and the culture and make a new model that's able to account for both. Okay, so I'm going to describe the sort of ins and outs of the model a little bit now. If anyone has any more questions on the sort of premise.
So I don't know if this is on the premise, but I wonder, is the fact that the population is getting smaller, is that helpful in terms of you get smaller and therefore a mutation would have an easier time going to fixation because it's smaller? Is it, although it's dangerous also, how does the size of the population play into these calculations?
Yeah, it's hugely important. Yeah, the size of the population changing. Sorry, I've just done something to my screen. Yeah. So yeah, absolutely. Actually, we're going to talk about that in a couple of minutes too, and that is very important to the interaction between the genes and the culture as well. So yes, as the population becomes smaller, the cultural traits and the genes can both spread more rapidly through the population, frequency-wise. Yeah.
I want to ask one more question about, this is unrelated. I mean, this is about just the rescue itself, the evolution rescue, just a genetic one.
Yeah.
But is this, does this describe COVID well, for example? Is that, can you say COVID has gone through population decline and then has a new variant and that's the population rescue through genetic variation? Or is this not a good model to describe COVID?
So the context that I've seen this in most often is in antibacterial resistance. So very often, and actually I think there are some models on HIV, but antibacterial resistance in particular is important because they're often like these like mutations that arise that allow the population to be rescued in like a particular individual. So when you're trying to kill the population of bacteria and they mutate and then they are able to rescue themselves. I don't know if I can say anything about whether COVID has done that or not.
Okay, thanks. Yeah.
But certainly bacterial and viral populations are like the target of this work predominantly. So there's no reason why not. Okay, so the model looks a little like this. Wait, yes. Okay, so the model is made up of individuals, and each individual is characterized by a phenogenotype. So this is sort of very typical for gene culture coevolutionary models. We describe a phenogenotype. It is a combination of a genotype and a cultural phenotype. Phenogenotype. So here, each individual has a diploid locus, which is a big A locus. The ancestral form of the allele at this locus is small a, its frequency is Q, and we also have a cultural sort of haploid locus. So the transmission, we're going to get on to in a couple of minutes, but the transmission is sort of separate for the cultural traits and the biological traits, genetic traits. The ancestral form of this is small b. And by definition, we say that this has low fitness in the new environment. So fitness low enough to cause population decline in the new environment. So the fitnesses are labeled W and with the phenogenotype. But you can imagine a different phenogenotype. So here we've got mutations. So here the big A allele is a beneficial mutation. Its frequency is P. The big B innovation is the beneficial innovation and its frequency is X. And again, by definition, this has a higher fitness in the new environment. Okay, so the absolute fundamental building blocks of this model are the fitness equations. So we want to describe the fitness of every phenogenotype, and there is a manageable number of phenogenotypes in this case, so we can put all these together, and everything that I do, all of this modeling and all of the analysis is based on these building blocks. So this phenogenotype, small a, small b, is ancestral for both genes and culture. This is the worst fitness-wise. They have a fitness of 1 minus D. And D describes the sort of severity of the mismatch between the environment and the genetic and cultural background. But everyone suffers from this mismatch between the environment and the genetic and cultural background, and we're interested in mutations that change that relationship. And so here, the big A allele confers a benefit HSA, where H determines the effect of a single allele as opposed to homozygote. So typically H is 0.5 for all of the analyses that I've done here. So this is just saying there's no sort of heterozygote advantage. This is then 2 copies of the big A allele. It confers a sort of typically greater benefit, again, if there's no heterozygote advantage, SA. And then we come on and we do the same thing with the cultural trait. So small a, big B has the ancestral genotype, but the novel innovation, and they have a fitness 1 plus SB. So we do the same for culture. B is the beneficial innovation, and it confers benefit SB. And we can fill in the rest of these in the same way. Okay.
So these numbers, SA and SB, are sort of positive numbers I should think of. But
sorry?
the SA and SB, I should think of these as positive numbers.
Yes, these are positive numbers. Yeah. Yeah. Exactly. Okay, so what can we do with these fitness equations? We need to know a few things in order to look at the probability of evolutionary rescues using these fitness equations. So we want to figure out how quickly the new beneficial traits spread on average and what the dynamics of their spread might be. So what are the differences maybe between a gene-only rescue, a gene culture rescue, or even a culture-only rescue, which is not a possibility that I'm going to talk much about today, but just we want to know what the differences between those dynamics are. And we want to know how quickly does the population die out, taking the presence of the new traits into account. So we need to recalculate the rate of decline of the population. And finally, then, with what probability can we expect the new traits to sort of beat the clock and rescue the population?
Can I ask about model assumptions real quick?
Yes, of course.
So I may be getting this wrong, but the way I understood the fitnesses is that culture and gene effects are sort of multiplicative with each other, right? So first of all, why? And second of all, why do the 2 gene effects, those seem to be additive? Or where I have 2 gene mutations versus 1 gene mutation, the difference was this H times SA. So I'm wondering why you picked these assumptions. How much could they be? How much consequence would they have?
If they were additive.
Yeah. So
I guess that, you know, so I have no real good answer for you. I mean, it's convenient. Is the honest answer, but what difference it would make? I think there would just be different constraints on the values of SA and SB. Because of course they need to fit within certain constraints. And as it is right now, they can have quite a wide range of values. They can take on a wide range of values. And it's easy to figure out, say, the relationship then between SA and SB that sort of gives us different dynamics. But I don't really have a good answer for you. It's just what we chose to do.
Right. I guess for general types of fitness effects, you can define SA and SB such that you parameterize these effects based on these two numbers. Maybe all you need is monotonicity of, okay, the gene effects plus the cultural effects are doing more than just either, and the two gene effects are doing more than just one gene effect. Okay, maybe that's the only thing that you might need. So, sorry, I'm just basically thinking.
That's okay.
Thanks.
Yep.
So are you going to assume that the arrival rates of these different mutations are much slower for genes than for cultural things?
Yes.
Yeah, okay. Okay, good, yeah.
Yes, I will assume that eventually.
If I may add one thing, so the model by Osmond and colleagues, that it assumes that it's really hard selection going on, and that fitness is directly translated to population growth rate,
Yes.
right? And therefore it's not really cannot easily be scaled away. So it's not a matter of scaling, and that you could easily step over to a fitness measure that is something like rank equivalent or something like that.
Yeah,
so
these are
it's
absolute
really
fitness.
And there I wonder a bit, because you're assuming then hard selection, whereas then in the context of cultural evolution, you typically have something, you are living then by definition more or less in a kind of a community of people, where then most of the selection processes are more governed by soft selection, so frequency dependent selection and these kind of things.
Sure, yeah.
Can your approach also cover these kinds of other processes?
Yeah, so I kind of touched very quickly on the fact that the transmission process for the genes and for the cultural trait are separate. So what happens first is that there's so individuals pair up and produce offspring, and the genetic transmission happens, and then there's another process of cultural transmission on top of that. So that's a naive population, and they then undergo sort of, well, it's very kind of simple oblique cultural transmission at that point. So you're right in the sense that the biological fitness values that I've shown are absolute fitness values, but those are not what determine the cultural dynamic.
Okay.
So they're sort of separated out, if that makes sense. But yeah, it's a good point.
Okay, thank you.
Sure. Okay, so yes, so the first things that we need to do then is to calculate the average biological fitness, that's W bar, and the average cultural fitness, V bar. So this is what we're going to use for the cultural change. Calculate the change over a single generation in the frequency of the beneficial allele, given that it exists. So this is all sort of given that the allele exists, what do we expect to happen to it? So that is delta P and delta Q. So we can use this for some numerical simulations. And we calculate the change in frequency, of the beneficial innovation, given that that exists. So this is delta X and delta Y. And again, we want to use that for our numerical simulations. We want to calculate how long it should take for a cultural trait to fix. followed by how long it should take for a mutation to arise and establish for our analytical results. We use these to generate a probability for the establishment of a mutation and the establishment of an innovation before extinction for a particular value of D, for a particular mismatch between the environmental shift and the genes. And this determines the sort of rate of population decline in the presence of these different forces. And we finally sort of put all this together to generate a sort of massive probability for the emergence and establishment of an innovation and a mutation, which is a gene culture coevolutionary rescue. So we're not super interested in the situations in which the rescues happen regardless of the culture, or culture is able to rescue the population on its own. We're most interested in this gene culture coevolutionary dynamic. So I'm not going to go through all of this today. I think that would be cruel. A lot of it is tedious, but I do want to flash up a few of the equations just so that we can kind of look at the equations, what they depend on, what their form is, so that we can sort of understand a little bit more about the results that I'm going to present. And again, it's all the results are preliminary. So okay, in order to look at the equations, I think first I want to just go through what the population dynamics look like. So how reproduction happens, both on a biological scale and on a cultural scale. And just to pull those things apart and to look closely at the parameters involved. So in order to do that, I'm just going to pull apart the phenogenotypes. I'm going to represent them, the genotype as this AA and the cultural phenotype in a small bowl underneath the individual here. I'm going to put them into a population. So this is a population of n individuals, or of 8 individuals, I guess, n equals 8. And the first thing that happens is biological reproduction. So the first thing that happens is that all of the individuals in here are paired up and they produce offspring in sort of a very standard kind of way. So they produce offspring in the way that you would expect them. The number of offspring that each individual is expected to leave is given by their WI, which again is dependent on the fitness SA, SB, the environmental shift, severity of the environmental shift D, and the heterozygote advantage H. And so they produce this group of offspring, which might be, you know, smaller in size than the previous generation, or might be bigger in size than the previous generation, but they're naive as far as cultural traits are concerned. So the next thing that happens then is cultural transmission. So this is, as I said, extremely simple. And the naive individuals look at the last generation and they say, okay, with some success bias or some sort of ability to tell what's a good thing, what's a bad thing, which I have labeled G, I decide which trait I'm going to adopt. And they adopt a cultural trait. And this is the full reproduction event. And this goes on for a number of generations. So the population might decline in size, it might increase in size, or it might stay the same. Okay. So again, a lot of these equations are kind of tedious to derive, and I don't want to do that, you know, live on a recording. So I'm just going to flash up these equations. So this is the equation for delta X. And so this is the change in the frequency of the cultural trait over a single generation. And what I really want to pull out of this is what it sort of depends on. So the first thing that I want to note is that the change in X depends on the frequency of the cultural trait. So it depends on X. It also depends on G. So it depends on how good we are, say, at identifying good traits or the strength of a success bias of some description. And it depends on the average cultural fitness or in the population. We can do the same thing for the change in the allele frequency over time. This is a little bit more complicated, but sort of rests on the same kind of assumptions. So what I want to pull out of this is that the change in the allele frequency over time depends on the frequency of the alleles themselves and also on the frequency of the cultural traits. So in here, there are, you know, P's, Q's, X's and Y's. So it depends on the frequency of all of the alleles, and it depends on the frequency of the cultural trait. It depends on all the fitness values that we calculated before. So these were biological fitness values. And it depends on the average biological fitness, where fitness is measured as sort of infant survival or fertility, something. The last thing that I want to show is this is sort of, once you put all of that together, this is the probability of a gene culture coevolutionary rescue. It depends on the rate of decline of the population and many other things. I apologize in advance for how this looks. But what, again, I want to pull out of these equations is what the probability depends on and in what way. So it first depends on, of course, the time to extinction when a cultural trait can arise. So this is different to the purely genetic case. This is the probability that a beneficial allele can arise and establish before a beneficial trait. So this is essentially negligible, this part. And finally, it depends on the probability that a beneficial allele can arise and establish after a beneficial cultural trait. So you can see in here, this quantity depends on the environmental, the rate of decline of the population, D, and now it depends on the rate of the decline of the population in the presence of the cultural trait. Okay. So the probability ultimately depends on the dynamics of the allele and on the trait, on G, so the success bias, on both the fitness benefits, SA and SB, on the initial population size, N0, and the initial rate of decline, D.
Okay. Excuse me, can I ask?
Sorry?
Yeah.
I saw just that you have the genetic mutation rate is probably mu in there.
Yes.
Do you also have something like a cultural innovation rate? Is it which parameter would that be?
Yeah. So for some of these, I have an innovation rate, and for some of these, I assume that the innovation arises more or less immediately after the environmental change. So it depends on whether you believe that it would take a very long time for a cultural innovation to arise, or whether you think that it would take less than a generation for a cultural innovation to arise. So in this one, there is no innovation rate. It's just assumed that the innovations arise almost immediately, I think.
Okay, thank you.
Sure.
I mean, I was also wondering a little bit what these, what mu, sigma squared, and epsilon and so were. So mu, I guess, is the mutation rate.
Mu is the mutation rate, and epsilon is some sort of function of the S, the selection parameters. So this is the expected number of offspring that an individual with a genotype leaves is 1 plus epsilon. So this is a function of the S parameters in here. And this is the sigma is the variance in offspring number in the population. T is time, N0 is the initial population size.
Okay, thanks.
Okay, so I'm going to just talk a little bit about the results. Some of this is speculative, so that is what it is. Okay, so the first thing that I did was, okay, I said, you know, one of the things that we need to understand before we do kind of a full analysis is how do the genes and the cultural traits interact with one another? So what effect do they have on one another, given that both are present? So the first thing that I did was to ignore waiting times. So I say there's an environmental shift, and immediately a mutation arises, and immediately a cultural trait arises. This is obviously very unrealistic. This is not what would happen. But the aim of doing this analysis was to say, okay, how do the genes and culture interact when both are present? And so we can understand a little bit more about what's happening in the full model. So we want to know sort of what the gene culture interactions look like in a declining and then growing population. So to do this, we compare a gene-only rescue, a culture-only scenario, and a gene culture model in some numerical simulations that are based on the model that I presented above. So these are the results of that. So along all these x-axes are time. In the first panel, this is the cultural trait frequency, and I'm comparing a gene culture scenario with a culture-only scenario. And the take-home, really, I think the most interesting results from this is that the presence of the genes tend to slow down the presence of the cultural trait. So someone mentioned earlier that there is an effect of population sizes, and that's exactly what this is. This is kind of an indirect interaction that's based on the fact that the populations decline and grow at different rates depending on whether the cultural trait and the genetic trait are present. So on average, the trait can spread more rapidly frequency-wise in smaller populations. And so the presence of the genes tends to slow down the spread of the cultural trait. The second panel then shows allele frequency on the y-axis. So this is the genetic. And here we compare a gene culture case with a gene alone case. And we see here that culture also slows down the spread of the genes. So this involves the same kind of interaction that we had before, so some indirect interactions, but it also reflects a more direct interaction. So for example, there are some extra benefits that are accrued to homozygote ancestral individuals who have a good cultural trait, as compared to, say, a heterozygote individual who has a bad cultural trait. So because of these extra benefits that are accrued to individuals with not so great genetics, through the cultural trait, big A can't spread as rapidly as it does in the absence of the cultural trait. And finally, down here, we have population size in the third panel. So here we can compare the population sizes, or the sort of trajectories of the population rescues in a gene culture situation, in a culture only situation where there is no rescues, just population size just declines much more slowly, and the genetic rescue alone. So here, the gene culture rescue in general, and this sort of generalizes, I think, across a lot of parameter space, the gene only rescue, when it happens, is faster. So the gene culture rescue is in general faster than the gene only rescue. So this happens because although, both of the two elements are spreading more slowly than they would in the absence of the interaction, the combined effect on fitness still rescues the population more rapidly. Okay, so another kind of interesting thing that came out of this analysis had to do with the threshold and the fitness values of the mutations. So when we're waiting for rescue mutations to happen, we're not just waiting for any mutation to arise, we're waiting for a mutation that's good enough to rescue the population and fully compensate for the environmental shift. So on the y-axis here, we have this required allele frequency, allele fitness to allow a rescue to happen. And you can see, so in this black line, in the absence of culture, this has a fixed value, of course. But when we have a cultural trait that's reached a high frequency in the population, depending on the fitness benefit of that cultural trait, so this is, again, absolute fitness, biological fitness benefit of that cultural trait, the required allele fitness for a rescue mutation declines, and it can decline, quite far. So this tells us that culture sort of helps rescues along in two ways. So first, we have more time available to wait for a mutation to arise. And second, when it does arise, it doesn't have to be as good to save the population as it would have had to be in the absence of culture. Okay, so the last result that I want to show is about the probability of rescue. So this is the very ugly big equation that I showed before. So on the y-axis here is the probability of a rescue before extinction happens. On the x-axis is the environmental mismatch. So the brown dotted line here is the probability of a rescue in sort of human-ish parameter space. And you can see that it's very low. It's not actually zero, but it's very close to zero. So in the absence of a cultural trait, the probability of an evolutionary rescue under these circumstances with these parameters is very low. And when we introduce the cultural trait, so one thing I just want to flag before I sort of show these results is that I've kind of stacked the deck a little bit. So when the environmental mismatch is 0.05, so just at the very end of this axis here, the cultural trait, if it appears in the population, will preserve the population for a very, very long time. So the probability of rescue before extinction at this point will always be 1. So everything's going to start up here and then we'll decline from there. So we're going to look at what happens when the success bias or the strength of cultural transmission is relatively low. And that's what happens. And then we can continue to look at stronger and stronger transmission. And this is what happens. And you can sort of take, reasonable parameters and say, okay, when culture is spreading very slowly or the success bias is not very strong, then although we have a slightly higher probability of an evolutionary rescue, it's still not really that high, all the way up to a very strongly transmitted, rapidly transmitted beneficial cultural mutation, which changes the probability of a cultural rescue from somewhere near 0 to somewhere near 40%. So, I mean, these are very extreme parameters, but. Okay. So this is just saying that the probability of true sort of evolutionary rescue increases when a beneficial cultural trait extends the life of the population. The faster the trait can spread, so the higher G, the slower the population decline, and the better the chances of an evolutionary rescue. And putting this all together, we sort of found that gene culture interactions increase the probability of a rescue, make it faster when it does happen. So there are more faster rescues, but the spread of both the gene and the trait is slower compared to situations where they spread alone. And this is controlled by a delicate balance of parameters. And so I just want to, so anyone who's sort of got a toe in evolutionary theory might at this point be wondering whether I'm just modeling a Baldwin effect, which is a totally fair thing to ask. I also don't mind if I'm modeling a Baldwin effect, but I think it's worth asking whether this is in fact a Baldwin effect or not. So the Baldwin effect is sort of notoriously, I didn't quite know how to describe this. So underspecified, misunderstood, or misused are very broadly used to describe a variety of interactions between learning, behavior, and genetic adaptation. So it's worth asking, because so much has been written and so much has been understood about different types of Baldwin effects, whether this is a kind of Baldwin effect. Of course, this depends on what definition you want to use. So in order to discuss this, of course, it's open to different interpretations, but I'm going to stick to the sort of original Baldwin effect. So particularly drawing on, I think it was Godfrey Smith, who wrote in 2003 this particular formulation of a Baldwin effect. So it's worth discussing whether this is in fact an instance of a Baldwin effect or not. So I'm just going to do that really quickly. So the Baldwin effect entails the following things. So the first thing is stage 0. So there's a new environmental condition which arises. Absolutely, we have modeled that, yes. The next stage is that natural selection favors learning as a consequence of changed conditions. Now, this is not what we model, so we're not looking at the evolution of the capacity for learning. We're not looking at whether natural selection favors an increase in innovativeness or capacity for social learning as a consequence of these changed conditions. We assume that those things exist already because we're looking at modern human populations. So we're not particularly interested in whether natural selection is favoring learning per se. We're more interested in whether cultural selection is favoring behavioral adaptations to changed environments. So this is not quite what a Baldwin effect involves. Stage 2 of the Baldwin effect is mutation, recombination, and selection, leading to the proliferation of genotypes that produce behavioral responses without learning. So this is the kind of genetic assimilation part of the Baldwin effect. this is kind of a little bit more tricky. So when I think about this model, I tend to think about something more like cold weather clothing, for example. So we know that Europeans, if encountered colder conditions than we were used to, and innovated warm clothing much, a long time before they had genetic mutations to compensate for cold, for example. But these are not quite the same things as what the Baldwin effect is talking about, because the genetic adaptations don't produce behavioral responses. So they are addressing the same evolutionary question, but they're not producing behavioral responses without learning. So they're not producing innate behavioral responses. So this one I think is a little bit ambiguous. I'm not 100% sure. I think that what we've modeled doesn't exclude behavioral responses, the genotypes producing behavioral responses, but it doesn't necessitate it either. And finally, stage 3, so this is sometimes included and sometimes not, so this might not be so important to think about, but at the end of the Baldwin effect, when all of this has happened, learning is no longer under selection. And so if it's costly, for example, to maintain an innovative brain or the capacity for social learning, it will diminish. And of course, we're also not dealing with that in this model. So we're still not dealing with the evolution of learning per se. So all in all, I think it's probably a little bit ambiguous whether it is technically a Baldwin effect or not, but I think on balance, it's probably not quite the same thing, maybe nuanced in a different way. Okay, so I don't know if we can stop here, or I can talk about stochastic simulations. I don't know what you guys would prefer, if you have questions or.
I would have I would be quite curious about, you say that you want to take this to data eventually, right?
I'll continue.
What the question I at least would have is like, how does this help you think about what data you might see and what not in the genetic makeup, I guess, of humans, right? Is that the idea?
That is the eventual idea. I'll continue because the next few slides are about that. So if that's what people are most interested in.
That's not just me, but yeah, I would
keep
be
talking. Okay.
I would also be interested in that.
Sorry?
I would also be interested.
Okay, cool. This is like the most speculative part, so I'm sure you'll be very dissatisfied at the end of this discussion. But anyway, I will continue. So the next steps that what we want to do is look at stochasticity and data analysis. So we showed that, okay, in theory, given some parameters, given some modeling assumptions, culture could make evolutionary rescue accessible for humans. But does this happen? And more importantly, did it happen in our evolutionary history? We don't really know. So we hope to use the model to figure out what differences exist between gene-only rescues, between selective sweeps, and between gene culture co-evolutionary rescues, should we be able to see these in genetic data. So there are existing descriptions of what gene-only rescues should look like. And we want to know, are there any consistent and potentially diagnostic differences between those rescues and the ones that we've described? We will maybe also think about whether, if we have cultural data, might we see some signatures in there, but I think that's even less likely. And I think there's a very real possibility that we will not be able to discern any of these processes in genetic data or in cultural data. But we want to develop at least tools that will enable us to find out whether it's possible or not. So the numerical simulations and the analytical models that I've shown you can only really tell us so much here. Stochasticity plays an enormous role in all of these processes. So the stochastic loss of novel innovations is a hugely important part of this, which is much more difficult to capture in numerical simulations and analytical models. So we have a stochastic agent-based simulation, which is able to do this. On average, so far, these agree with our analytical results, but of course, the devil is always in the details of these things. So the details of the stochastic loss and the patterns of stochastic loss are hugely important. So I'm going to move to completely speculative work now. So this is what we're working on right now. And I'm just going to talk really quickly about what an ideal outcome from this analysis would look like. So what we could possibly get out of an analysis like this. Okay, so most of the analysis that's been done on the signatures of selection, or the signatures of a population rescue through a selective sweep, involves some sort of pairwise distance. So some measure of heterozygosity, basically. So I'm going to show you some plots from Osman and Koop, and then talk about maybe where the gene culture coevolutionary model might fit in to that work. So they take a stretch of genomes. So this is what they model. And they have one locus, which is under selection, and then they have a stretch of DNA either side of that locus, which looks a little like this. So this is the one that's under selection right here, and these ones over here are less affected by the selective sweep. These ones over here are less affected, and as you go closer to that locus, you're more likely to see an effect or a signature of the selective sweep in the genetic data, and this depends on the recombination rate. They look at mean pairwise diversity across this stretch of DNA. And they say something like this. Okay, so if you're over here, you're very unlikely to be affected by the selective sweep. You're still affected by a bottleneck, but unlikely to be affected by the selective sweep. So your mean pairwise diversity might be up here somewhere, and ditto on the other side. This is sort of symmetrical. And then once you look at a sample down here where the locus is under selection, you get a dip in pairwise diversity. So this is sort of a signature of either a selective sweep or a bottleneck. This is reasonably well understood. And you can draw a line between these things. So this depends. So the sort of shape of this thing depends on recombination rates largely. Okay, and they get something that looks like this. So this is from their papers. This is from Osman and Koop 2019, The Genetic Signatures of Evolutionary Rescue by Selective Sweep. It was on bioRxiv. I think it's published in Genetics now, so it's probably best to look for that published version. But basically, this is exactly what I just said. And distance from the selected site is over here. This is 0, so this is the selected site. And then either side of that, and you get these characteristic patterns. And what they compare is a situation where you have a constant population size. So this is just a selective sweep. This is the genetic signature of selective sweep of this strength. And this is the genetic signature of an evolutionary rescue. And the question that we want to ask, I guess, so there's two things that I would say about this. So the first thing is that it would be very cool if we could put gene culture co-evolutionary rescues on this plot. And where on this plot it would end up, I'm not sure. So just to be completely speculative, I could imagine a situation where with a gene culture co-evolutionary rescue, the bottleneck is less severe than for a genetic rescue. So because the genes and the culture are acting on the population size, the bottlenecks might be less severe. So you might expect our line to end up somewhere in here. But you could also imagine situations in which the bottleneck is more severe because there are rescues where there wouldn't otherwise have been. So this mean contains only evolutionary rescues. And if we include gene culture co-evolutionary rescues, there will be more of them, and they'll be rescued from lower population sizes than was possible before. So it could end up down here, and I really don't know what would happen. My intuition sort of fails me on this, to be perfectly honest. So cool as it would be to be able to add a gene culture line to this plot and to be able to compare these things, we also have a bigger issue here. So if you're talking about bacterial populations in the lab, maybe you would be able to tell this pink line apart from this blue line. But for humans, I think this is considerably less likely. So we don't have hundreds and hundreds of repeats of human evolution, and something that you might have noticed if you're sort of looking closely at this plot. So these lines are all individual simulations that the variance here, so the sort of distribution of effects here are completely overlapping. And so the probability that if you took one of these thin blue lines, so one particular simulation, and you asked, well, which dynamic did it come from? Did it come from this one or did it come from this one? The likelihood that you would be able to say which one it came from, I think, is very low. And that's going to be a serious problem for us. So one of the things that we want to do, of course, is to replicate this, this work, to put our own work sort of in comparison with these things, but also to try and think of novel ways to look at genetic data so that we will be able to take the quality and the kind of data that we have when we look at gene culture coevolution, which is definitely not of this sort of quality, and see if we would ever be able to tell which processes have happened in the past. So one of the big questions we're asking is, how useful is any of this, given the spread and the overlap in all of these simulations, and can we do anything about that? So I think that's everything that I had prepared to say to you. Oh, I don't know. Yes, that's the answer. I really have no idea. This is all work in progress, and hopefully we'll be able to come up with something interesting. Let's see. Thank you.
All right, thank you very much. Clapping emoji, and I will stop the recording, and after that we will have about 15 minutes of open discussion.